So now let's take a look at our types of passive transport. And remember that passive means that we do not need ATP. So we do not need this energy molecule in order for these processes to happen. So we'll start with simple diffusion and then talk about facilitated diffusion and then osmosis. The first type of membrane transport that we're going to talk about is simple diffusion. And simple diffusion does not require ATP, so remember that this is a passive process. And the solutes are going to move down or with the concentration gradient. So remember that this means that the solutes are going to move from an area of high concentration to an area of low concentration. So if we have a high concentration area here, those substances are going to start moving towards the lower concentration area. Now, in simple diffusion, this refers to solutes or substances that can cross the phospholipid bilayer, meaning that we do not need a transmembrane or transport protein in order for this to occur. So remember, we have our phospholipid bilayer here, making up the majority of our cell membrane. So in simple diffusion, materials can go right across that phospholipid bilayer without any help. Now, simple diffusion also refers to once stuff is kind of inside the cell, that material is going to start spreading out. So it doesn't always involve crossing a cell membrane. Sometimes it just means that it's going from a high concentration area to a lower concentration area. So let's take a look at some of these molecules or substances that can cross the phospholipid bilayer unaided. So this means that we don't need those transport proteins. So remember that things that are lipids or lipid soluble are going to be able to cross the cell membrane without any help. Also our gases, so things like oxygen, and remember oxygen is O2 and carbon dioxide is CO2, and those can go right across the cell membrane without any help. And then other very small substances are going to move across the cell membrane using this simple diffusion. So if it can cross the phospholipid bilayer, then it can move by simple diffusion. This picture is showing you simple diffusion happening across the cell membrane. So you can see in the picture that we're kind of looking at what's happening over a period of time here. So we start out with a high concentration of whatever this substance is on one side of the cell membrane, and then a low concentration on the other side. And what's happening is that these little molecules are vibrating and they're bouncing off of each other. And as they bounce off of each other, they start to bounce away from each other. Now, if this is a substance that can cross the cell membrane, as they bounce off of each other, they're going to bounce across that cell membrane. And so more and more of these are going to be moving from the area of high concentration to an area of lower concentration. Now, in this last panel, kind of down here, this is what happens when the cell reaches that equilibrium that we talked about. So equilibrium means that the concentration of these molecules on either side of the cell membrane are the same. So there's no longer a concentration gradient. And so those molecules are going to kind of stop moving across the cell membrane. Now, they're not going to stop completely, but we're going to end up with a one-for-one exchange, meaning that for each one of these molecules that moves across the cell membrane in one direction, a molecule is going to move in the other direction as well. So equilibrium is where the concentrations are the same, so there's no longer a concentration gradient. And there's going to be no net movement of substance. So that means that that's that one-for-one exchange in which one molecule might cross the cell membrane, but for each one that goes in one direction, there's going to be a molecule going in the opposite direction. Interestingly, substances are going to diffuse according to their own concentration gradient. So if we look at this picture here, we see that we have these kind of purple or reddish circles here, and these kind of yellow or orange circles here. And for the red molecule, the high concentration area is on the left and the low concentration area is on the right. But for the yellow molecule, the high concentration area is on the right and the low concentration area is on the left. And so what we see happening over time is that the red substance is going to diffuse across, or I'm sorry, it's going to diffuse down or with its own concentration gradient. So it's going to go from the left to the right, whereas the yellow substance is going to diffuse down or with its own concentration gradient. So they're both diffusing by simple diffusion, but they're going down or with their own concentration gradients. Sorry, just make sure you can see that one there. Now, this happens, for example, at the lungs. So at the lungs in the body, there's typically an exchange of oxygen and carbon dioxide that are diffusing, I should say, in opposite directions. So oxygen from the lungs might be diffusing into the bloodstream, whereas carbon dioxide from the bloodstream would be diffusing into the lungs. As I mentioned on a previous slide, simple diffusion does not have to happen across the cell membrane. It can also refer to solutes just spreading out in a solvent. So in this example here, we have some sort of dye that is being added to a liquid. And the area where this dye is added is a high concentration area. And then you can see over here that there is a low concentration of those dye molecules. Now, over time, those molecules start to spread out, and they start to move from that high concentration area to that lower concentration area until they are evenly spaced throughout the solvent. So we see this also if you were to, for example, spray some air freshener in a room. When you spray the air freshener, there would be a whole bunch of that air freshener right where you sprayed it. But over time, those molecules that smell good, hopefully they smell good, are going to start spreading out in the environment until you can smell it everywhere in the room. The next type of membrane transport we're going to talk about is facilitated diffusion. First of all, facilitated means with help. So if you're facilitating something, you're helping to make it happen. So in the case of facilitated diffusion, the helper is going to be one of our transport proteins. So one of these transmembrane integral proteins that is crossing the cell membrane. Now these proteins here may be called one of these things, and it kind of depends on a little bit what their location is, what their structure is like, and what's crossing them. But you'll see these terms used in a couple of different places. But anytime you see the term channel or carrier or pore, it's quite possibly talking about our facilitated diffusion. And as a reminder, proteins are very specific as to what they can transport. So a transport protein for sodium is going to be different than a transport protein for glucose. And then you need a different protein to transport fructose. One kind of cool thing about these channels or these transport proteins is that sometimes they can be opened or closed. So some of them are always open. So if they're open, it means that the material can cross from one side of the membrane to the other side. But in other cases, they might be opened or closed. So if we look at this example here, this is showing a closed transport protein. And then after something, in this case a ligand, binds to a receptor, as we kind of mentioned before, it opened that transport protein, which is kind of like unlocking a doorway so that this molecule can now move across the cell membrane. And they can be gated or, I guess, locked or opened by different things. So, a ligand gated channel is something that is opened by some sort of chemical bonding to a receptor. So, we typically see these with things like neurotransmitters and hormones. So, these are how neurotransmitters and some hormones work. In voltage gated channels, there's going to be a change in voltage or a change in charge that happens that opens or closes a one of these channels. And we also see this in things like neurons and in muscle cells. So these are how neurons and muscle cells kind of transmit the information needed in order to either communicate if it's a neuron or to contract if it's a muscle cell. Mechanical gated channels are ones that we find in some sensory receptors. So for example, the sensory receptor for hearing are called these hair cells. And hair cells are mechanically gated. And what happens is that sound waves kind of move into the ear, and these sound waves kind of physically bend these structures. And when it bends these structures, it opens up a channel and it allows something to cross that channel. And that's how the information gets kind of converted from sound waves to a signal that your brain can understand and process. Now, facilitated diffusion has some similarities to simple diffusion. So just like simple diffusion, it is passive transport, which means that it does not require ATP, which means that it's not requiring energy in order for it to occur. Also, like simple diffusion, during facilitated diffusion, solids are going to move with or down the concentration gradient. So this means that those substances are moving from a high concentration area to a lower concentration area. Now, one main difference between facilitated diffusion and simple diffusion is the substances that are crossing the cell membrane. Because remember, these are substances that require a transport protein. So things that don't require a transport protein are going to be able to cross the cell membrane by simple diffusion. But things that require a transport protein are going to travel by facilitated diffusion. So these are things like our ions and other charged particles. Remember that other charged particles include things that are polar, which means that they're hydrophilic, which means that they are either attracted to water or water soluble. It also includes these large molecules as well that just can't fit across those phospholipids. So it can't fit across the phospholipid bilayer. So instead, it needs help, and it gets help through a transport protein. Now we're going to move on to talking about our types of active transport. Now, there was a type of passive transport that we haven't covered yet, and that's our osmosis. So remember that this is a type of passive transport, but I'm going to talk about it at the end of this PowerPoint because it's kind of its own special mechanism. So we'll talk about that a little bit later. But right now we're going to focus on our active transport. And remember that the main thing about active transport is that it requires ATP. So it requires that energy in order to occur. Now we're going to look at kind of the two main types, and then we'll also talk about another type that we kind of call vesicular or bulk transport, which includes endocytosis and exocytosis. But it also is kind of its own, it's also its own kind of process or mechanism. So we look at active transport, primarily looking at these two types here first, and then we'll talk about how vesicular transport is kind of its own thing, just like osmosis is. So, so first of all, regardless of whether we're talking about our primary active transport or secondary active transport, one thing to keep in mind is that it's similar to facilitated diffusion in that we need this transmembrane protein. So remember that these are the transport proteins. So just like facilitated diffusion, we need some transport proteins. But the big difference between active transport and facilitated diffusion is that these solutes are going to move up or against the concentration gradient. So remember that in facilitated diffusion, the solutes were moving down or with the concentration gradient. In active transport, the solute is getting moved against its concentration gradient. So let's start by looking at primary active transport. And so we're going to focus on a specific example known as the sodium-potassium pump. And one thing that you'll notice is that with active transport, or especially primary active transport, the word pump is just another name for the transport protein. So the proteins are called pumps. Now with the sodium-potassium pump, it's got some specificity, but it's actually specific to two ions. So it's specific to sodium and potassium. And if we look at our example here, for each ATP molecule that is expended in this process, there are going to be 3 sodium ions that are transported in one direction across the cell. And there are going to be 2 potassiums that are transported in the opposite direction. So for each molecule of ATP, you're transporting 3 sodiums and 2 potassiums, but you're actually transporting them in opposite directions. But they are both being transported up or against their concentration gradient. So potassium is being transported up its own concentration gradient. Sodium is being transported up its own concentration gradient. So what we end up with is a lot of sodium on one side of the cell and lots of potassium on the other side of the cell because of this pump mechanism here. And this actually sets up a concentration gradient across the cell membrane for both of these ions. So there is a sodium concentration gradient and a potassium concentration gradient that's being set up across the cell membrane. And this is important because this becomes the driving force for secondary active transport. Vesicular or bulk transport is a type of active transport. So remember that that means that we do have to have ATP in order for it to occur. And it involves the use of vesicles. And vesicles are basically like bubbles of the cell membrane. So part of the cell membrane can actually pinch in order to form a vesicle. So we can kind of pinch in like that, and then it can bud off. And so you essentially end up with a phospholipid bilayer forming a bubble. And so in vesicular transport, there's something that maybe is outside the cell that gets kind of sucked into the cell, and then it's contained within this vesicle. Or it might be going in the opposite direction. So sometimes it's something that's inside the cell that gets transported outside of the cell. Now, one kind of interesting thing is that as you can see here, the substances that are getting pulled into these vesicles, they're not coming in contact with the intracellular fluid. So they're actually in their own kind of self-contained vesicle here. There are different types of vesicular transport, and we can divide them into essentially two kind of main categories. So there's endocytosis, which means that something is being brought into the cell. And exocytosis means that something is leaving or being sent out of the cell. Now we can further break down endocytosis into phagocytosis and pinocytosis. And phagocytosis is considered cellular eating, meaning that whatever is being brought into the cell is something that is typically big and solid. Pinocytosis is where a liquid is being brought into the vesicle, but the concept is pretty much the same in that something is being kind of the opposite of what we're seeing in this picture here. Something is actually being brought into the cell and being pulled into one of these vesicles. Now, exocytosis is just something that is inside a vesicle that is being sent out of the cell.